Okay, hello everybody, good afternoon. Welcome to the HRM seminar for today, May 18th, 2021. It's a pleasure for us today to have as our seminar speaker, Dr. Matthew Anderson. Dr. Anderson is Associate Professor of Pathology, specializing in Neuropathology, and Associate Professor of Neuroscience at Harvard Medical School. And he's the Chief of Neuropathology in the Department of Pathology at Beth Deaconess Medical Center in the Boston area. Dr. Anderson received his bachelor's at Cornell College in Iowa City, Iowa, and then his MD PhD at the University of Iowa College of Medicine in medicine, physiology, and in biophysics. And then was a resident at the University of Iowa Hospitals and Clinics in anatomic pathology. In the late 1990s, he joined Harvard Medical School, Brigham and Women's and Children's Hospitals as a clinical fellow in neuropathology. And at the same time was a research fellow at MIT in neuroscience and immunology. And over the last 15 years or so, he's been chief of neuropathology at Harvard Medical School, BIDMC and Children's Hospitals. It's really great to have him with us today. The title of his talk is going to be Genetic Autisms, Behavioral Circuit Mapping, and Targeted shRNA Therapeutics. So Dr. Anderson, thank you for joining us, and the floor is yours.
Great. Thank you so much, Frank. It's a wonderful chance here to share our work. And, certainly the RNA or oligonucleotide therapeutics is making a major impact in some of the neurodevelopmental disorders in the very near future and epilepsies. And I'll show you kind of some examples that might be ultimately targeted. So autism is a behavioral disorder. It's defined by behaviors, and that's really the subject of my talk. Its incidence is climbing. I think it's even higher than what I have here. It's like 1 in 43, I think now on the CDC. Male predominant over female, still don't understand why that is. It's heterogeneous at the genetics level, but genetics really only accounts for about 15% of the total cases. And at least in this talk, I'll be using some of the dominant genetic forms to map out the molecular pathways and circuit mechanisms that underlie the phenotype, the behavioral phenotypes. The phenotypes come at a very early age, in early childhood. It's required for the diagnosis. They have impaired sociability and social communication. They have increased repetitive behavior and restrictive interests. And there are major comorbidities that often can be some of the most debilitating problems, such as irritability, tantrums, and aggressive behavior. And I'll talk more about that in a little bit. They can have epilepsy, which can be mild to quite severe and sometimes can lead to premature deaths. And I'll show you how epilepsy can interact with some of these other behavioral phenotypes. They have variable amounts of intellectual disability as well. So you can see the clinical syndrome is, easily could be very heterogeneous, but I will show some examples of convergence in genetic pathways where multiple genes seem to hit the same circuits to affect sociability and this irritability aggression phenotype. And I'll show you some examples where epilepsy converges as well. So the irritability phenotype displays at all ages. Here's a child which is, having a tantrum. Here's an individual that's being restrained from self-injurious behavior. And then here's an individual who's grown up quite large now, and they become more and more difficult to manage. And you can see the damage to his room that has happened from this behavior. And this can lead to pretty major problems in the family with crisis intervention referrals. Sometimes the family can't handle it themselves, and it requires residential placement. It's one of the few things that medications have been approved for in autism, and it's, I'll talk more about those in a minute. Certainly a poor quality of life for both the individual and the parent. So risperidone is one of the mainstays for treating the aggression phenotype. These others are also effective. Interestingly, they all have this comorbidity of weight gain. And as I described for you, the circuitry we think is involved, keep this weight gain phenotype in mind. We're also going to provide some, at least preliminary evidence for a site where risperidone may be acting to affect this phenotype. So as I said before, it's very genetically heterogeneous, but which only accounts for a small fraction. And you know, autism really is a fairly new disorder, and we're really just beginning to fully understand it. This, as recent as 1997, this was kind of the cutting edge at that time of genetics and autism, and it was a cytogenetics abnormality where there is inherited from the mother extra copies of the chromosome 15 region. And it was interesting that it was inherited from the mother in this example. In a more contemporary 2009 paper using CGH arrays to look at gene copy number variations across the genome, here again, this 15q region was a major hit again in 2 different cohorts you can see, and there were no examples in the control cases. In this paper, they also show some other genes, which I will highlight later, that converge into a molecular pathway. We actually decided to pursue autism focusing on this genetic subtype. And what's convenient is there's a whole range of gene dosage changes of this locus, from interstitial duplication to an extranumerary isodicentric chromosome here. You can see in a cytogenetic spread to deletions. And these are all inherited from the mother, which implicates a imprinted gene in this locus that's responsible. And I'll show you what we found to be important in a minute. There are some other disorders which are inherited from the father when it's deleted as well. So there's a variety of imprinted genes in this locus. What is known about this locus is that there is a gene called UB3A, and I'll talk about what it is in a minute, but maternally inherited mutations of UB3A produce Angelman syndrome, which is a neurologic disorder with intellectual disability, motor defects, some seizures, severe language deficits, but they have a kind of an unusual personality where they smile and laugh a lot and have what appears to be almost a pro-social behavior. And as I said, that's really the opposite of the impaired sociability that's typical of autism. And this IDEC15, which has this triplication of the maternal copy of this 15q1113 region, also contains this UB3A gene. And I'll show you in a minute, but it's imprinted. So it's only expressed from the maternal copy in neurons. So if you delete that maternal copy, you've lost all of UB3A. If you'll notice here, there's sort of a reciprocal behavioral phenotype where hypersociability, at least that's how we interpreted this, contrasts to the impaired sociability. So we thought that maybe the same gene in excess doses might be underlying the impaired sociability. There's some reciprocity in the irritability behaviors as well. So this locus has many genes, but the only gene that's consistently imprinted such that it's expressed exclusively from the maternal allele is this gene UB3A. So UB3A is a multifunctional protein, first really described as a E3 ubiquitin ligase that degrades P53 by adding ubiquitin tags, which drives it to the proteasome for degradation. But it's also a transcriptional co-regulator, which only partially depends on its E3 ligase activity. And it's co-activator for progesterone, estrogen, androgen, glucocorticoid receptors. It's also called E6AP. That's what's seen here. And the imprinting you can see in this panel, where the paternal allele expresses a long non-coding RNA, which silences the paternal copy of UB3A in neurons. And interestingly, a recent study targeting, this RNA molecule, in fact, was aimed at Angelman syndrome, where the UB3A gene is defective on the maternal allele. And the general idea was to desilence this UB3A gene so that you might get a recovery of the protein's function. And in fact, it was quite successful. So that is a really nice example of a long non-coding RNA leading to imprinting specifically in neurons and targeting that long non-coding RNA as a therapeutic using oligonucleotides. So we thought that UB3A might be the key, so we really focused on that gene specifically. To model autism-like phenotypes, when I first got into this field, I wondered if one could even measure such things. But in fact, mice are very sociable animals, just like humans are. And you can monitor the amount of time that they explore a cage that containing a conspecific versus the other cage, which is sort of just a novel object over here. We also measure ultrasonic vocalizations when mice are paired, and we can also concurrently, when they're put together, measure their physical contact. We also have a method of monitoring for aggression where a male that's sexually mature and with a female partner in its home cage, you take the female out, introduce a male intruder here, and the mouse that's the home cage mouse will attack that intruder. It's sort of it's not an abnormal phenotype. It's somewhat protecting its own territory, but we can look at the level of aggression in that circumstance. So we actually, acquired, or in this case, generated mice that have 1 extra copy, 2 extra copies, or a loss of the maternal UB3A gene. Remember, losses are connected with what looks clinically like hypersociability. And here is the protein levels. You can see a knockout, maternal knockout versus 1 copy versus 2 copies of our back full-length transgene. And sure enough, we showed in this study here that these mice are in fact hypersocial in the paradigms that I'll show you in a minute. This genetic interstitial duplication is really not that strongly penetrant, but there are individuals designated with autism that have this. So there may be some other environmental or genetic interactions that happen to make this phenotypic. And I'll talk about that in a minute, a little later. But these mice were fully impaired in their sociability and also had repetitive behavior. And I'll show you later, they also have aggression. So we now have a tool with which to really explore the mechanisms of these phenotypes coming out of particular brain circuits that might underlie sociability or aggression. So we wondered what cellular process might be important. I mentioned that UB3A is an E3 ligase, and it certainly targets a variety of proteins in the cytoplasm and in other compartments for ubiquitination and regulation that way. But it's also it's a transcriptional co-activator. So we actually, this study is published, if you want to see, the first half is published work, the second is unpublished work. So we wondered maybe this nuclear effect of UB3A that had been studied mostly in the cancer world might be involved in how its actions on neurons. So we actually took our full-length back construct, which we had flag tags on the C-terminus, and introduced nuclear localization signals as a fusion protein as well. Made more mice, and conveniently we had mice that had the nuclear targeted in 2 different independent founders at a dose of protein and mRNA that was equivalent to a mouse with just 1 copy. And remember, this is not phenotypic, and I'll show you that in a minute. But so therefore, if you target it to the nucleus, can you make a mouse that's not phenotypic with this dose of protein phenotypic in these behavioral assays? Here is the staining of the flag antibody against the untargeted, just the general UB3A back transgene versus the nuclear target, and you can see it co-localizes perfectly with the DAPI signal. Doing staining, you can see the dose of 1x here in the nuclear fluorescence intensity is much higher in the NLS targeted, which actually approaches the 2x copy amount. We also did biochemical studies showing enrichment as well. So here is the 3-chamber sociability task where mice explore the two sides. And you can measure various things. One, I'm just showing an example of is sniffing time, this chamber versus this chamber. And you can see that even wild type mice that are littermates of the 1X mice show a really predominant time sniffing the social stimulus. The 1X mice don't really show a phenotype, whereas 2X mice do. They're impaired in their preference for social stimuli, a little bit like we might expect in an autism model. We have a flag tag version and an untagged version. We get the same impairment and sociability in both. Here is the nuclear targeted version, which actually has the same protein and mRNA dosage as the 1X, which has no phenotype, but now the nuclear targeting fully impairs their sociability. And as I mentioned, mice with a knockout of UB3A are hypersocial, and you can sort of get a trend of effect here. But if you look at prolonged sociability, they just continue to be very hypersocial. It's not really well appreciated here. We also, as I said, measure ultrasonic vocalizations in paired females. If you put two males together, they fight, so we focus on the females. And at least in this genetic form, it's equally penetrant in males and females. So you can see a lot of vocalizations when wild types are paired and in 1X. So again, no impairment with 1X UB3A dose, whereas the 2X with the flag or the untagged impair their social vocalizations. And having a dose of nuclear targeted UB3A that's equivalent to the 1X does now impair their social vocalization. And here you can actually see the hyper sociability phenotype where the vocalizations when the mice are paired are strongly elevated. And interestingly, the knockout phenotype resembles the phenotype if you socially deprive a mouse. So if a mouse has been single housed for like 12 to 24 hours, they will become hypervocal and hypersocial as well. So this really argues that UB3A is acting in the nucleus to impair the 3-chamber sociability and the social vocalizations. We also found that mice in cages with these, UB3A mice had injuries in the tail. They had bite wounds, and in their hind limbs they had injuries as well. And sure enough, if we did the 3-chamber, resident intruder task measurement, the 2X had increased attack time and attack number compared to the controls. And the 1X in this case, just like with the sociability, were not statistically significant, although you can see a bit of a trend here. So we think that this model has both the impaired sociability and the increased aggression phenotypes. Does the nuclear targeted UB3A increase the aggression as well, like it had was responsible for the sociability? And again, we use that same tool I just showed you. And sure enough, despite having similar protein and mRNA levels as 1X, these nuclear targeted and this not producing an increased attack, the nuclear targeted versions did produce statistically significant increases of attack behavior. So we think that both phenotypes, both behavioral phenotypes are coming out of a nuclear action of UB3A. But as I'll show you later, we think that those two phenotypes map to different brain circuitries. Okay, so we have an action of UB3A acting in the nucleus to produce both of these major autism-related phenotypes. How are we going to find the molecular pathway beyond that? We at least now know that it's probably gene regulation, because that's one of the major actions of any molecule in the nucleus. So we decided to look at what genes are regulated by UB3A in the brain. And we compared the wild type mice to the 2X mouse here, on a genome-wide basis. And then we took all these tools that we had and did gene expression analysis by qRT-PCR. And you can see here the 1X versus the 2X. There is a dose-dependent mRNA, as we saw for protein. And here is one example of the nuclear localized, which has the same mRNA levels as the 1X. And this is just the untagged here. The knockout, maternal knockout, it completely abolishes most of UB3A. There's a little bit in some of the endothelial and astrocytes that's not imprinted. This is one example of a gene that was repressed by increased UB3A. You can see that it's halved almost, maybe 40% decrease with one copy of UB3A, and it was significant. Two copies really strongly repressed it, as did the nuclear targeted. So here, just like both the behavioral phenotypes, when you target UB3A to the nucleus, you can actually achieve kind of the same level of impairment as you get with 2X. And here is the reciprocal change, as we saw on the sociability, a reciprocal increase in sociability. We have a reciprocal increase in cerebellin expression. So if you looked at all the genes that we saw regulated, it was quite a intimidating set of genes. We saw approximately 400 that showed a really nice dose-dependent nuclear-based repression by increased UB3A in the cortex. We didn't know which brain region to look at the time because we didn't know where the behavioral phenotypes mapped. You could see increases here in the knockouts. We saw about 200 that are upregulated, and they had some dose dependence, but not as cleanly as the downregulated genes. All right, so how do we tackle this? How do we find the key targets that might be involved in a molecular pathway for autism? And how do we understand what UB3A may be acting on to impair sociability or increase aggression? Well, we actually took advantage of the wonderful, so that's the goal, is what genes are targeted by UB3A to lead to these phenotypes. We took advantage of a ever-growing network of autism genes that have been uncovered through extensive characterization, through exome sequencing and other methods. And we created a protein-protein interaction network between those and the UB3A regulated genes. The edges here in blue represent protein-protein physical interactions, which we started with a more stringent approach to the protein pathways. And then we did cluster analysis to find tightly interacting clusters of proteins. And you can see we found like 12 of those here. Then we took and applied gene ontology classification to ask what might those clusters be doing. And this turns out to be a gene regulatory cluster with many transcription factors. This is a glutamatergic synapse cluster. This is a developmental cluster for the nervous system. This is protein ubiquitination. This is a phosphorylation pathway, including mTOR, MAPK pathway, cell migration, so on and so forth. Okay, so that really does help at least organize the data set. Now, the other thing that I mentioned is that there are certain genes that seem to get hit over and over again. UB3A is at the very top of the list. And also naturally, a major way that you could impair behavior would be to impair synaptic transmission in circuits that underlie those behavior. So we look very closely at the glutamatergic synaptic transmission cluster, and conveniently we found the second most common copy number variation in autism, Neurexin 1, or at least in some case sets, was physically interacting with cerebellin 1 and postsynaptic. That's a presynaptic molecule. This is a postsynaptic molecule, GRID1. I'll show you a little bit later what that is. But this forms a transsynaptic complex. This UB3A targets cerebellin 1. This is a secreted protein that physically binds the pre and postsynaptic membranes to actually organize the synapse. So we thought that that was a kind of a major candidate target whereby UB3A might be producing these behavioral phenotypes. Here is UB3A in this paper, and here is Neurexin 1, the second most common and the second strongest p-value. And you can see down here is GRID1, which is a known binding partner for cerebellin 1 as well. And in the case of UB3A, as I showed you before, there's increased copies which repress cerebellin. So there's a loss of cerebellin when UB3A is high. Well, these are both deleted in autism, Neurexin 1 heterozygous and GRID1 heterozygous. So we think that there could be a critical synapse somewhere within the brain that is organized by a Neurexin 1, cerebellin 1, GRID1 transsynaptic complex that might underlie autism-related behavioral phenotypes. And I'm going to provide the evidence for that next. Okay, so in a more recent analysis, you can see UB3A is one of the more common duplications, and Nerexin-1 and GRID1 are deleted, at least in the family of these various molecules. Cerebellin-1, it has actually a GWAS signal that's not really well represented in the CNV data set here. It's also heavily expressed in the cerebellum, and that's really where it was first discovered to be a synapse organizing molecule. And when you delete cerebellin, you're actually going to get a severe ataxic phenotype as well. So it may be the cases where cerebellin is deleted are classified instead as ataxia, maybe with some autism symptoms, but we haven't really fully resolved that. So cerebellin is not throughout the brain. It's in the granule cells. That's where it's got its name of the cerebellum, cerebellin. And it's in the entorhinal cortex that connects to the hippocampus. It's in strategic parts of the hypothalamus we'll talk about a little bit later. It's in the midbrain, and including an area we'll talk about later. It's in certain hypothalamic nuclei. So it's really uniquely in certain spots, not everywhere. It's also interesting because like increased UB3A, increased neuronal activity itself can repress the expression of the cerebellin gene, and that's just shown here in a paper that's been published. KCl depolarization of the granule cells that have a lot of it, or canic acid in vivo can repress the levels of cerebellin expression. Why is that important to autism? Well, activity is one of the, seizure-related activity is one of the things that you find in autism. If you look at cases collected of early infantile epileptic encephalopathy, where that's the focus of the genetic study, you find these genes mutated. But independently, if you collect cases for autism, many of the same genes are also found mutated. So there's really an extensive overlap between these two disorders, suggesting a possible causal link. So we wondered whether seizures in our hands could repress cerebellin-1 and concurrently impair sociability. And ultimately, I'm going to get to, whether, cerebellin has a causal role there. So we use the seizure paradigm where we use a chemoconvulsant injected on a daily basis intraperitoneally, and we monitor the seizure severity. And there's this phenomena of plasticity of the seizure severity with the same dose of chemoconvulsant where it gets worse and worse and worse. By day 10, you're up here. We looked 24 hours after this day at the level of cerebellin mRNA in strategic brain regions, the cerebellum, the medial amygdala, hippocampus, entorhinal cortex, ventral tegmental area, and VMH. And you can see cerebellin was repressed. And concurrent with that impairment, we had impairment in the social behaviors that you typically connect to autism. Impaired sniffing in the case here, but chamber time as well. I just don't show it throughout. And the vocalizations were also impaired. So the seizures repress cerebellin while impairing the same social behaviors I was describing earlier. Now, UB3A also impairs sociability, and we wondered if seizures might act through UB3A or synergize with UB3A to act on these phenotypes. And therefore, we took the knockout mouse, the Angelman's mouse, and remember, they're a bit hypersocial. We hit them with seizures versus the wild type. And you can see here that this 10 daily seizure protocol severely impairs sociability in the wild type, but the maternal knockout of UB3A is resilient to that. And as is the repression of cerebellin resilient now to that. So it suggests that UB3A may be in the pathway. We also have these 1X mice, which have no impairment of sociability. We think that this models the sort of interstitial duplication dosage of that disorder. We've wondered why, only a subset of the human patients are phenotypic there. Maybe there's some other genetics or some other, comorbidity. And certainly you do see seizures in this disorder variably in patients. So we used a milder version of the seizure protocol. You can see the kindling curve here in wild type and 1X mice. And that was not enough to abolish the sociability in the wild types, but it was fully impairing sociability in the 1X mice. So you do have sort of a synergy at this level and a protection at this level. And here again, you get a synergy in repressing cerebellin using seizures plus a lower dose of UB3A. Now, this leads us to a model where all these conditions are converging on each other. We have UB3A repressing cerebellin, and we have Neurexin-1, which is a known binding partner of cerebellin-1, and GRID1, also a binding partner. And we have seizures, which hit here as well. So we wondered, could seizures not only synergize with UB3A, as I showed you, but also with GRID1? And that is what I show here. Here's a GRID1 heterozygous mouse, only kind of mildly attenuated sniffing, probably not significant. If you give a mild seizure protocol, the wild type is not impaired, whereas the GRID1 heterozygous mice are becoming fully phenotypic now. What if we bypass the UB3A and the seizure and just directly delete cerebellin-1? Will that impair sociability? The answer is yes. If we do it in the glutamatergic population, you can see a full impairment of the sociability, phenotype, as well as the ultrasonic vocalizations in a male paired with a female or two females paired. So we wondered which neuronal circuits might underlie this effect of UB3A and seizures, and whether these might, act on that circuit to repress cerebellin to produce these phenotypes. So we made another tool. We took that full-length UB3A gene with a flag tag and introduced a lock-stop-lock sequence so that we could turn it on at will with different promoter Cree lines. You can see here this transgene with the flag tag, and we had the stop cassette in there, and you could clip it out with Cree to express this flag tag version, extra copies of it. And you can see here the glutamatergic population are pretty broadly distributed, and the GABAergic population are far fewer. And if there's no Cree present, not shown here, there's no expression at all of that flag that's bound to the protein. So we now had a tool. This tool did not express at as high level as the 2X mouse, just to tell you now. So we actually took this tool and we crossed it to glutamatergic, GABAergic, kind of cortical, subcortical, a subset of subcortical, dopaminergic, noradrenergic, cholinergic, and serotonergic Cree lines. So we really fully mapped the gamut of cell types in the brain. At baseline, they were not impaired in any of the examples, but as I said, the dosage is probably closer to a 1X. When you hit them with a seizure, though, we found phenotypes in the glutamatergic population and the dopaminergic population only. So we think that the seizure UB3 synergy is happening at a dopamine and glutamate population. So in the brain, the dopamine is extremely well known to be in this mesolimbic circuit that is involved in reward and substance abuse and things like that. It's in the midbrain. So and there's also a glutamatergic population in that same location. So we actually honed in directly on this ventral tegmental area that is involved in reward. And we focus on the glutamate cells because that's where cerebellin loss actually impaired sociability. So we introduced this Cree conditional version of an AAV viral vector carrying a double inverted LOX-P flanked UB3A. It's a particular isoform, and it's injected stereotactically in the ventral tegmental area in either VGLU2 Cree or wild type mice. And this just shows in a knockout mouse that we can get really nice expression, and it's in both the nucleus and in the cytoplasm, this particular isoform. Sniffing at the baseline was only partially attenuated, much like we had seen in the other example previously. But when we gave the mild seizure protocol, we got a full impairment. So in this very specific brain region, in this very specific subset of cells in that location, adding UB3A, we're able to get a full-blown impairment of sociability in that location. we wondered if cerebellum loss in that location itself was sufficient to impair sociability. So we took a mouse that had a LOX-P site flanking critical exons of cerebellum 1, introduced AAV viral vectors carrying CRE or just GFP into the ventral tegmental area. That's the GFP is shown here in that region. It's expressing nicely in neurons. Sure enough, no seizure was necessary. Now we could completely abolish their sociability. And by contrast, if we cross this flock cerebellum mouse to dopaminergic neuron CRE, there's no sociability deficit. So we think that the cerebellum is acting in the glutamatergic neurons, but not the dopaminergic neurons, and particularly in the ventral tegmental area, this sort of reward substance abuse circuitry. Although this glutamatergic population is one that has really not been linked in any way to reward, or, well, recently it was linked to reward, but not so much substance abuse. We know that cerebellum is a synapse organizing molecule, and it seems to be critical in these glutamatergic neurons. There is a circuit involving glutamate neurons that projects from the brainstem to this kind of striatal circuitry. So we wondered if the cerebellum was organizing a synapse in that circuit. So we introduced channel rhodopsin, which allows us to shine light on the axon terminals in this part of the brain here while doing our intracellular recordings of the neurons in that location. And here you can see VGLU2 with the channel rhodopsin has a very nice light evoked synaptic response. That's a excitatory synapse. But if you delete cerebellum 1, it's abolished. And here is the statistics from many cells. We also, instead of deleting cerebellum everywhere in all the glutamatergic neurons, we did it selectively in the VTA region. And here you can see a mouse that has channel rhodopsin activated by CRE, or channel rhodopsin activated by CRE plus a deletable cerebellum homozygous. And here again, only intervening on the cerebellum in that location was sufficient to fully impair those synapses as well. So we think that cerebellum is acting on synapses that are glutamatergic. We wondered if we took away the glutamatergic transmission from the VTA, whether that would impair sociability, because we just saw cerebellum loss nearly abolishes synaptic transmission. And what we found was, yes, indeed, deleting glutamatergic synaptic transmission by eliminating VGLU2. It's the vesicular glutamate transporter that sort of loads the vesicles with glutamate in the synaptic terminus. That does impair sociability, whereas there is a subset of cells in this midbrain region that co-express dopamine and glutamate. The neurotransmitter complexity of that location is quite high, and this is something that was relatively recently discovered. Well, deleting in that dopaminergic population that has VGLU2 did not impair sociability. So we think it's the glutamatergic population, not the dopaminergic population, or at least the co-expressing subset. So, this was a completely novel population of cells that people had really not linked so much to behavior. So we wondered whether activating these cells might actually drive sociability, or inhibiting them might abolish sociability. And there's this wonderful tool or technology called chemogenetics, or DREAD, designer receptor exclusively activated by a designer drug. I truly believe that this may become a major therapeutic approach in the central nervous system. You'll see the amazing effectiveness of this method to turn neurons on and off. And I think also certainly in other organ systems in the future. There are muscarinic acetylcholine receptors that couple to GQ or GI that are activated by clozapine N oxide, CNO, and when this receptor is present, it will, elicit a signal transduction pathway that ultimately leads to increased neuronal firing, whereas this one will affect other signaling pathways that decrease neuronal firing. And there's a kappa opiate receptor activated selectively by salvinorin B through mutagenesis that we also used here. That's inhibitory. So that's actually what I'm showing here, VTA targeted injections of a Cre-activatable core, the inhibitory one here, expressed selectively in glutamatergic neurons. And just adding DMSO, the carrier for salvinorin B, you can see really strong sociability. But if you add salvinorin B, you abolish their sociability completely. If we do the reciprocal, where we activate those neurons in the VTA that are glutamatergic, here's just a saline injection. They normally are sociable for the first 5 minutes, and then it's lost. They sort of attenuate their interest in social stimuli, and the Angelman's or the single house mice are quite sociable in the second 5 minutes, but normal wild type mice are not, including these mice that we injected. But if we add the activator of this receptor that couples the GQ to activate neuronal firing, we can get very hypersocial mice that remain sociable into the second 5 minutes and possibly beyond. And you can see a bit of a dose response curve here. So certainly if we activate these neurons, we can get them, make mice sociable, more sociable. If we inhibit them, we can take away all their sociability. What if we applied that to the seizure UB3A synergy impairment? That's shown here, where we co-inject UB3A along with the chemogenetic tool into the VTA VGLU2 cells. And at baseline, there's normal sociability. We hit them with 5 seizures. In the saline mice, there's no sociability, whereas we add just even a small dose of CNO and we fully recover their social behavior. Same with just adding the dread in this region. And now, instead of introducing the UB3A seizure synergy with the sort of alternate paradigm, we just hit them daily with the 10 PTZ protocol, and that abolishes sociability. But if you add the CNO back to turn on those cells, you can fully recover sociability despite these seizures having probably impacted all, very many circuits throughout the brain. Now, what about the cerebellin? Is that really the key molecule in this UB3A seizure synergy? So we actually created a viral vector that would have a Cre-activatable cerebellin in it. We added UB3A and cerebellin, or just GFP, into this location. You can see both baseline sociability was normal. You hit with 5 PTZ seizures, and the GFP mice are impaired in their sociability, just as we saw before the seizure UB3 synergy phenotype. But if you add extra cerebellin there, you can actually make them resilient to that seizure-induced impairment. So we really truly believe that of all the molecules that UB3A is regulating within these cells, this is truly the key molecule that might underlie the impairment of sociability in that circuitry. So this, I'm not going to belabor this, but we are beginning to try to understand more about these particular cells and the circuits involved in social behavior. What about that aggression phenotype I started with? Can we use these same tools to map that circuitry out? Cerebellin is in the ventral medial hypothalamus of the, somewhat uniquely in the brain. And this circuit had been shown previously to be a locus where you can make a male mouse attack a female mouse, which is almost a pathologic version of aggression. So we had that sort of information in the back of our minds as we were starting to map out where aggression comes from. We found that VGLU2 targeted increases of UB3A elevate the attack behavior. We found that VGAT population does not, so it's probably a glutamatergic population. This is the ventral medial hypothalamus, and this is the ventrolateral subdivision, and that's really where previous studies had implicated attack aggression behavior phenotype. SF1 is a transcription factor that is in VMH very strongly, and yes, when we overexpress UB3A in those cells, that's enough to increase aggression. We also introduced our Cre-activatable virus into selectively the ventrolateral subdivision of VMH glutamatergic neurons, and that was enough to increase attack behavior. So we think that excess UB3A in that location might be doing, creating this phenotype. Really, to know that that is the site as opposed to elsewhere, we wanted to ask if we could rescue the aggression by removing the excess UB3A from that location. So we made instead of a Cre-activatable, we made a Cre-inactivatable UB3A back transgene where LOXP sites flanked critical exons and made transgenic mice, which had a baseline increased aggression phenotype. And we targeted Cree virus to the VMHVL now. Instead of overexpressing, we deleted the UB3A that's there. And sure enough, we could reverse the phenotype in adult mice. So this really argues that this phenotype, despite this defect having been there throughout their life, that could be reversible. So the circuits are not in a sort of aberrant developmental state, but kind of still plastic in adulthood. So we wanted to understand the specific neuronal subtypes that might drive aggression in the VMHVL. Some people had implicated TAC1 in Drosophila, so we thought that those might be key. We used a implanted mini microscope where mice can walk around carrying this microscope, and had a very long grin lens that went all the way down to this area, so we could measure calcium signals in the TAC1 neurons. Here's cell 1, 2, and 3. And you can see when we introduce a male intruder, you can see a strong activity in response to that intruder, and also corresponding to events of attack. So we know that these neurons are active during attack. Are they sufficient to drive aggression using this chemogenetics tool? There's also a progesterone expressing subset in that same location, and activating dreads caused a tremendously intense attack behavior in both these subtypes of neurons in the VMHVL region. We could introduce excess UB3A selectively in that TAC1 or PGR population and increase aggression. And we could actually even use a dread to silence that aggression, the inhibitory dread as a tool. Is cerebellin responsible in this location as well? And I showed you that cerebellin is heavily expressed there. Well, sure enough, NLS down regulates cerebellin in that location. That's the nuclear targeted version of UB3A that has a lot of aggression, I'll show you. If we delete cerebellin in the SF1 Cree mouse, that's a transcription factor in VMH, somewhat selectively, you get increased aggression. If we inject a Cree only in the VMHVL in the flock cerebellin, we can get increased aggression. And if we cross to a TAC1 mouse, we can get increased aggression with deleting cerebellin only in that subpopulation of cells. And then to spatially constrain this intervention, we introduced a nuclear targeted version that we had also made into the VMHVL TAC1 Cree mouse, and only in those particular neurons. This produced a very intense aggression, and that was rescuable using this AAV-DIO cerebellin that can be, substitute any cerebellin that might be repressed. And you can see that we were able to, again, a little bit like the ventral tegmental area, the sociability phenotypes, we're able to, relatively robustly rescue the aggression phenotypes. So we think among all those potential UB3A targeted genes, when it's acting in the nucleus, cerebellin might be the key at this location as well. And remember, we got to cerebellin through this transsynaptic complex that involved other autism genes. So as I showed you before, there is some seizure synergy with some of these other molecules. What about in the aggression circuitry? We think that Norexin is acting maybe in a presynaptic site, grid on the postsynaptic site. And sure enough, you delete Norexin and you get increased aggression. You delete grid 1 fully and you get increased aggression. And actually, we found a synergy between the 1X and these heterozygous dosages, which you found in the human population, suggesting kind of a polygenic mechanism where in the context of a 1X UB3A, heterozygous Norexin produces increased aggression, as does grid 1. So I just got a few slides. I'm almost done here. So that's the candidate location we think is important for grid 1. And we targeted grid 1 deletions to a subpopulation, the AGRP population in this arcuate nucleus. And not and it doesn't work in VGAT population to increase aggression. And we did stereotactic injections of here's some shRNA example, grid 1 that's creactivatable as a shRNA using a TADA-LOX, lock-stop-LOX version of the virus. And in the arcuate AGRP neuron, but not in the POMC neuron, we get increased aggression. So we think that this is the postsynaptic binding partner for that phenotype. Sure enough, there is a synapse from the VMHVL to that location. We found that deleting the other genes that increase aggression do break that synapse, so we think it might be the critical one. And when we add cerebellin back, that synapse is rescued. So we think that that might be the key synapse. And actually, AGRP neuropeptide Y cells in the arcuate are the feeding driving neurons. When we turn them on, we also see repression of aggression. Here's attack with the dread that activates those neurons, and aggression is attenuated. And when we inhibit them, they have an active role in repressing aggression. And then finally, this is, I think, one of the last bits here. We're actually introducing excess neuropeptide Y. We can repress aggression. That's one of the peptides that those cells express in the GRID1 knockout or in the 2xUB3A mouse. And we can actually introduce risperidone. I mentioned that very early on as the mainstay in therapy for aggression in autism. That represses aggression in the 2X mouse, kind of a little bit analogous to the human situation. And all these interventions enhanced a feedback inhibitory loop. So there is a feedback inhibitory loop. So this has this collateral synapse that activates this cell. And this, we think, is a feedback inhibitor to repress aggression. So if you add neuropeptide Y, which represses the aggression phenotype in these genetic autism models, particularly in those cells, we can elevate this feedback inhibitory loop. And surprisingly, if we add risperidone systemically that represses aggression, we also magnify that feedback inhibitory loop. So what we did was we actually took multiple autism genes to assemble a transsynaptic complex and molecular pathway and use those tools to map down to a particular circuit in a particular part of the brain where we think that the aggression phenotype arises. In support of that notion, we find that risperidone magnifies that feedback inhibitory loop. So it's really a broken synapse that activates a feedback inhibitory loop. We think that the synapse going out to drive aggression would be intact. We haven't measured that yet. So our lab is generally looking at these autism genes as a tool to map out irritability circuits, sociability circuits. We could do epilepsy. We haven't really been doing that. Self-injurious behavior may be related to aggression, but it may be another circuit. And certainly there's a comorbidity of depression, which we've thought about sometimes. And we think that these approaches really lend themselves to thoughts around gene therapy using viral vectors, chemogenetics as a therapeutic tool to up or down regulate the circuits. And then finally, as you can see, these things are plastic and they're reversible in adulthood. So I think one could imagine a wide range of RNA-based or other therapeutic approaches to these circuits. So I think I will end there. Thank you.
Great, thank you very much, Matt. That was extremely interesting. We have time for some questions. So if you'd like to unmute yourself and ask a question, or you could sort of type things into the chat. I'm going to start by just asking one of the questions that's in the chat from Sergi. He asks, you are pointing at cerebellin as a possible effector of UBL3A in your model, but are you aware about examples of cerebellin dysregulation in Angelman patients?
Recently, somebody did provide pretty good evidence that a subset of Angelman syndrome phenotypes are coming out of an action of UB3 in the nucleus. So we think that it might have relevance to the deficiency of UB3A state. And we haven't yet made that connection, but certainly there's a lot of cerebellin in the hippocampal entorhinal cortex, in particular superficial layers. And that circuit is involved in, we think, intellectual disability. So cognitive impairments would potentially arise from deficits in the hippocampus. So it could very well be that, in the case of Angelman syndrome, the absence of UB3A you saw made them resistant to seizure-induced downregulation. Well, maybe physiologic plasticity, when circuits are active, utilize this regulation of cerebellum
刚
method.
刚我这里完了，还有3分钟。
So
Yeah.
we haven't solved it yet, but we suspect it might be true.
So there's another question here from Lisa Hall. Do you have a theory on why there is a huge increase in autism, I guess, currently in society?
Interesting question. So we have a brain bank. We're part of a program called Autism BrainNet, and we collect controls and individuals that have passed away with autism. And I'm actually the nationwide neuropathologist for that program. And I've actually looked at many cases now, and we published a paper showing, because, the puzzle is what accounts for the remaining 85% of autism that is not explained by genetics, or at least these penetrant genetic mechanisms. Well, surprisingly, we found lymphocytic cuffing around blood vessels in a very patchy, scattered manner. They're mostly T cells, and they're cytotoxic, CD8 positive. And we also see a cytotoxic debris coming from the astrocytes at the glia limitans, sort of right at the barrier between the CSF and the brain. So we suspect that there might be another mechanism that might involve the immune system in a fairly large chunk of autism. So, if you think about any immune disorder set, there is a rising incidence of asthma, there's a rising incidence of a variety of immune disorders. It's certainly just speculation on my part, but I wonder if it may have some relationship to kind of the growing incidence of immunity. And then how do you get from an immune disturbance to some of these molecules and circuits? Well, we think that it might involve cytokines or something that may impact neuronal gene expression through similar pathways, we speculate.
Very interesting, thanks. A question from Yanyu Chen. Besides risperidone, would Abilify do the same trick?
Abilify, yeah, good
Abilify.
question. Yeah, it might. It's kind of surprising how many therapeutics that are effective for aggression also are promoting an increased feeding and sort of moderate obesity. And at least the ones which have that feeding impact, we suspect may converge with the risperidone mechanisms that we're uncovering.
Any other questions? Well, I'll ask you one that was going through my head as you were going along, and then you addressed it in the last few slides by sort of showing that some of the drugs that are used to treat human patients had some efficacy in your models. But what is the efficacy of these drugs on other behaviors, not just the aggression, but does it change the social behaviors in those mice? Is there any correlation between the human studies and your mouse studies?
In the social behavior realm, there is no medication. And at least what I showed you was there's a broken synapse onto a feedback inhibitory cell, the neuropeptide Y AGRP neuron. And we think risperidone is acting there to repress aggression. The social circuits, we haven't really found the postsynaptic binding partner, target yet, but we have the tools with which to do that right now. I think that may be a different mechanism. And I think that the drugs that are going to be effective for sociability will, hopefully be we may gain some insights through mapping that postsynaptic target. So I don't think it's going to explain, and that may explain why risperidone is primarily used for that. It's also used for things like schizophrenia. So, who knows how that is working. There may be some analogies there but we haven't really explored that in depth. But unfortunately, the brain is so complex and the circuits are so specialized. I think that it may be hard. I mean, certainly if UB38 becomes really a central player in other forms of autism, you might target that therapeutically.
Yes, it seems to me you have a nice animal model to start testing other neurological drugs. I mean, given all of the overlaps between some of these syndromes, I mean, have you started testing glutamatergic inhibitors and that sort of thing? Maybe at low doses, because you don't want to, affect too many phenotypes. But
yeah, no, I agree that it's really a robust system. We know know a lot about what's happening there. So I think one could start to explore a variety of FDA approved compounds in the system, and you could even do things kind of at a circuit level, like in these slice recording studies, because behavior just takes so many animals, but I've thought about high throughput systems that might allow us to get some hits and then bring it up to the behavioral testing.
all right, yeah, this is very interesting. I think we're going to end it here. Well, unless there's any other questions.
Can I ask a quick question?
Sure.
Professor, you're using the risperidone, which is a dopaminergic, but in your early talk, you just focus on the glutaminergic instead of dopaminergic. How could it that?
Yeah, traditionally it's thought of as a dopaminergic mechanism, but in fact, it's not specific. It also hits histamine and a variety of other receptors. So at least the way whereby risperidone increases feeding, there is a little bit of literature on that. And it's not dopamine in that case. It seems to be in other receptors that it might also cross-react with.
I understand this. Why couldn't we use the olanzapine or some other, the newer generation of antipsychotic to test? Because I fully understand that most of the anti-epileptic drugs are pretty dirty. They are multi-receptor targets.
Mm-hmm.
So could we use a more pure, specific, doper ligand versus a glutaminergic ligand to make a difference, other than that, I throw something into the dish and, if they get something respond, I can, demarcate it or tease out, is this mechanism a mix, doper, glutam, or, either one?
Yeah, no, I agree that the next phase in this research would be to try to understand within this cell. We have a circuit now, and we could certainly explore receptors and the mechanisms precisely and get something much cleaner than risperidone. I do believe that is feasible now.
Yes, and also the risperidone, they get the mice, obese, and they get fatter, and they gain in weight like, we prescribe those medication to patients, and they complain they gain 30 pounds, 40 pounds.
Well, that's honestly, that's wonderful to share. Thank you. That's exactly what led us to think about the arcuate nucleus, because that is the site, and the AGRP neuron, neuropeptide Y neuron. If you activate those cells, you can dramatically change feeding. What we hope is that there's a subset of cells that are AGRP neuropeptide Y positive in the arcuate, only a subset that provide this feedback inhibition of aggression, and a different subset that promotes feeding. And there are tools now. There are CFOS reporters that could be temporarily turned on and off right in the windows of behavior that really help you dissect out the subset of cells in a particular nucleus that are involved in feeding versus aggression, for example.
I'm not surprising, because that the VPM, and also the satiety center are around there. The
Absolutely.
animal, they get aggression when they get hungry. They get more aggressive. They attack. So it makes a lot of sense. So you stand back, look into the big picture. But in your picture, you didn't have the GABA receptors.
True. I mean, and VGAT itself didn't work. So I think that they may not be GABAergic, in fact, this peptide population that's critical.
Yeah, probably indirectly. Thank you.
Yeah.
I think we're going to end it there. Thanks everybody for your attention, for your questions, and thank you, Matt, for a great talk.
Thank
I
you,
hope you have some luck with the new RNA medicines that are showing efficacy in the brain, and hopefully the patients will benefit soon. Okay, thank you, everybody. We'll see you at our next event. Bye-bye.
Thanks so
Thank
much.
you.
Bye-bye.
Bye.
Thank you very much. Bye-bye.